Hello students, welcome back to our channel Lagace Academy. In this video, we are going to discuss on the topic deviation from Mendelian Law. Before going to the video, if you don't watch the video on Mendelian Law, please do watch the video and the video will be mentioned above. There are two types of deviation from Mendelian Law. One is allelic interaction and other type of deviation is gene interaction. In the case of allelic interaction, this interaction is between alleles of single gene and it is of two types, incomplete dominance and codominance. In the case of incomplete dominance, when heterozygous condition or dissimilar condition come together, the dominant allele fail to mask the recessive allele completely. The example of this dominance is four-o'clock band. Here, when a homozygous dominant red color plant cross with homozygous recessive white color plant, we will get a capital R small r, which is F1 generation, pink color plant, pink color flower for this plant. And when we cross this F1, then we will get four types of progenies that are capital R, capital R, capital R small r, capital R small r, and small r small r. Here one red, two pink, and one white. Even though there is a heterozygous condition capital R, that do not completely mask the small r, that is white color, so a intermediate color that is pink is developed. So here for the F2 generation, we will get a ratio of 1:2:1 of its phenotype. In the case of codominance, the two alleles express together and the dominant never mask the recessive one. The example for this codominance is ABO blood grouping. In ABO blood grouping, when a blood group person that we can write as IA, who is homozygous dominant, cross with B blood group person that is IB, also a homozygous dominant, then they will have a progeny that is IB. Here dominant of both the blood group are being present, so here both the blood group dominant present and both are expressed, then we can have a AB blood group. And these two are the examples of allelic interaction. Now moving to the next interaction, that is gene interaction or epistatic interactions. The interactions between two or more genes that affect the phenotype of the trait they influence. When two genes interact, one gene show masking the other gene. The masking gene is epistatic gene and the gene get masked is hypostatic gene. And there are of 5 types, that is co-epistasis, recessive epistasis, dominant epistasis, duplicate recessive interaction and duplicate dominant interaction. In the case of co-epistasis, there are 2 types of gene and the 2 genes can mask each other or have equalability. The example for co-epistasis is chicken comb pattern. When 2 patterns, that is rose pattern which cross with a pea pattern, and the expression of the rose pattern is capital R, small p, cross with small r, capital P, we get a progeny of capital R small r, capital P small p, which is a pattern of walnut. So when two walnut pattern, that is two F1 patterns are being crossed, then we will get of progenies as of two dihybrid cross, that are four types. 9 of them will be of capital R or small r, capital P or small p. 3 of them will be of ratio capital R, here small r or capital R, then 2 small p conditions. Then 3 will be of 2 small r and capital P, and this one will be capital P or small p, and one condition will be small r, small p. And in this condition both are capital, so as above mentioned it is walnut pattern. And this condition here, this capital R mask the small p, so here the condition will be rose, and here the small p, capital P mask the small r, so here the condition will be p type, and here both are recessive, so this type is single type. Okay, to confirm this crosses we can have a test cross. So when we test cross the recessive one parent with the F1 generation, that is capital R, small r, capital P, small p, cross with recessive parent or recessive one, capital r, small p, then we will have like 1:1:1:1 ratio, that is all the type will have one each individual. In the case of recessive epistasis, in this type, when a particular gene becomes homozygous recessive, it masks the other gene if even it is dominant. The example of this trait is of coat color of mice. In coat color of mice, A allele represent yellow color, B1 represent for black. So capital A, capital B condition, a gout colored mice cross with small a, small b, that is albino type mice, we will have a condition of F1, that is capital A, capital B, small b, that is a gout type mice. Then we cross F1 self, that is capital A, capital B, small b, cross with capital A, capital B, small b. There are 4 type of probability as mentioned above. 9 type will be capital A, smaller capital A, capital B, smaller capital B. 3 types will be capital A, smaller capital A, small b, then 3 types will be small a, capital B, or small b or capital B, then 1 type will be small a, small b. The first one shows a gout color. The second one, in the case of second one, usually need to show yellow color, but here the B gene which mask the A gene because it is in homozygous recessive condition. So here the color expected is yellow. Here the color expected is yellow, but due to the masking the result will be albino. and here the capital B condition, so it is black color, and here the small a, small b, then it is albino. So the ratio we can rearrange to 9:4:3. That is a gout 9, then 4 albino, then 3 black. When we conduct a back cross or test cross, in this case, so we cross the gout heterozygous gout with the albino, like capital A, capital B, small b crossed with small a, small b, we will get ratio of 1:2:1. Here 1 is a gout color, 2 is albino color, then the other one is black color. In dominant epistasis, here the heterozygous or dominant condition of a gene masks the other gene. In the example for this condition is summer squash plant, and when capital W comes, it is for white color and it is the epistasis gene, and Y allele represent for two colors. If it is capital Y, it is yellow color, and if it is small y, it is green color. Then white plant, that is capital W, capital W, capital Y, capital Y. Even though it is capital Y, because of this capital W, it masks the capital Y, cross with a plant that is green. Small w, small w, small y, small y. Then we get an F1 of capital W, capital Y, small y. It is also white because this capital W masks this Y. Then if we self cross F1, that is capital W, capital Y, small y, cross with capital W, capital Y, small y. As mentioned above, there are 4 type possibility. 9 types will be capital W and small y or small w or capital W, capital Y, small y or capital Y. Then 3 types will be capital W, small w or capital W, small y, and then 3 types will be small w, capital Y, and the other one will be capital Y or small y, and the last one will be small w, small y, small y. For the first color it will be white because the capital W masks the capital Y, and this one also will be white because the capital W masks the small y, and here the small w, but here y is capital, so the color will be yellow, and here all are small, so the color will be green. So we can rearrange this ratio to 12:3:1. Here 12 color will be white, then 3 color will be yellow, then 1 color is green. Okay, then we can have a test cross to show its back cross ratio or test cross ratio. For that we can test cross the heterozygous white color plant with green color plant, that is small w, small y. Then we get of the ratio 2:1:1, where 2 color are white, then 1 color is yellow, then the other color is green. For a duplicate recessive interaction, for X to become Z, both A and B allele is needed. If only A present, only intermediate product is formed. So, it means that for a trait to be expressed, both the allele to be dominant. The example for this trait is snail pigmentation. So, a pigmented snail crossed with an albino, that is capital A, capital B crossed with small a, small b. So the F1 is capital A, capital B, small b, which is a pigmented one. Then this F1 are self-crossed for F2 generation. Then capital A, capital B cross with capital A, capital B, small b condition. So there are four possibility. First one, capital A, smaller capital A, capital B, small b or capital B. Third one, three of them will be capital A, small or capital A, small b. Three of them will be small a, capital B, small b or capital B. Last one will be small a, small b. In this, only first condition, both the alleles are indominant. So, first condition will be pigmented. Here, only A is dominant. So, no pigment, that is albino. And here only B is pigmented, so this one also albino. Here nothing is pigmented, is dominant, so that is also an albino. So the ratio we can reconstruct as 9:7. Okay, so when we cross this heterozygous condition with a complete homozygous recessive one, that is for test cross or back cross, small a, small b condition, we will get 1:3 ratio. Here 1 is pigmented and the other 3 is albino. The last one is duplicate dominant interaction. In this, for a thing to get a product, that is X to become Z, or an trait to be expressed, both the allele are not needed. Any one of them only needed to be homozygous dominant or heterozygous dominant. Okay, the example for this condition is shepherd purse seeds. There are two types of seeds. One is triangular shape and other is top shaped. If a triangular shaped seed, any A or B become homozygous dominant or heterozygous dominant, it will be triangular shape, and if all are recessive, then it is top shaped. So we can cross a triangular shaped, that is capital A, capital B with top shaped, that is small a, small b. We will get a capital A, capital B, small b, which is a triangle shape F1. Then this F1 is self-crossed, that is capital A, capital B, small b, crossed with capital A, capital B, small b. then we will have four different possibilities, that is capital A small a or capital A, capital B small b or capital B, capital A small or capital A, small b, small a, capital B small a or small b or capital B, small a, small b, and this will be triangular shaped because both are dominant. Here A is dominant, so it is also a triangular shape. Here B is dominant, so it is also a triangular shape. Here both are recessive, then it is a top shape. So we can change the ratio to 15:1, that is 15 triangular shape and 1 top shape. So when we cross for test cross, capital A, capital B small b, cross with small a, small b, we will get ratio of 3:1, that is 3 triangle shape and 1 top shape. And that's all about the deviation of Mendelian law. If you like the video, please like, share and subscribe our channel and follow us on Instagram. Lagos Academy, your educational catalyst. Please stay tuned.